Welcome dear students. In this video I'll be going to talk about the third step of glycolysis, that is the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. As you can see, the third step is actually irreversible step. So the first step is irreversible, second step is reversible, third step is also irreversible, and the name of the enzyme that play a role, that is called Phosphofructokinase1. In short, you can remember PFK1, Phosphofructokinase1. This enzyme performs this function. But as I have mentioned in my first video, that those reaction, there are 10 enzymes responsible for glycolysis. So there are basically 10 steps. Among them, 3 enzymes are rate limiting enzymes. That means they perform this process irreversible in manner. Like the hexokinase, one example is actually a rate limiting enzyme. The same is true for phosphofructokinase1, PFK1. So I will make a star mark for this enzyme. You must remember. Another important fact is this is the principal rate limiting enzyme. Principal rate limiting enzyme. Principal rate limiting enzyme. Okay, that means this enzyme is also go through allosteric regulations, and this enzyme perform major functions to control the glycolysis in a cell. Okay, now I will talk about how this enzyme is allosterically regulated. At first, I will show you some example of allosteric activator and also allosteric inhibitor of PFK1. The example of allosteric activators like adenosine monophosphate, adenosine diphosphate, and another very interesting is that fructose 2,6-bisphosphate is also a allosteric activator of phosphofructokinase1 enzyme. Allosteric inhibitor like ATP and citrate. This step, you can see here that another phosphate group is added. I have mentioned fructose 6-phosphate. Let me draw the structure of fructose, then you can understand. So the fructose 6-phosphate look like this. Here CH2O PO3 minus 2, then OH, then here AOH H. This is actually opposite. Then it is HOH. Then there will be H, and there is a CH2OH. You can see here, this is called the fructose 6-phosphate. Sorry. Here will be the CH2OH. Let me draw another time. CH2OH, and here will be the PO3 group. That means phosphate group at the 6th carbon, PO3 minus 2. Why? You can see here, first, this is the first carbon, second carbon, third, fourth, fifth, and sixth. Now this is the fructose 6-phosphate. Now this fructose 6-phosphate is converted into fructose 1,6-bisphosphate by phosphofructokinase1, PFK1. Why it is called bisphosphate, not diphosphate or biphosphate? The reason is, if there is any gap between the phosphate group among the carbon, then it is called bisphosphate. You can see here the phosphorylation occurs in the first carbon and the 6th carbon. That's why it's called bisphosphate. If the phosphorylation occurs in the first and second, or so on so forth, 2 or 3 or 3 or 4, so on so forth, then it is called biphosphate. Okay, as the phosphorylations actually happening in a distant carbon, it is called bisphosphate. So when this enzyme convert this product like fructose 6-phosphate into fructose bisphosphate, it actually simply adds another phosphate group to this molecule. Now the source of this of this phosphate group is also like the ATP. So here ATP is important. ATP associated with magnesium ion is converted into ADP, and it actually donates the phosphate group. Okay, now there is activator and inhibitor. So if I draw the enzyme, overall enzyme structure, let me draw like this. Let's suppose this is a PFK1, phosphofructokinase 1. In the PFK1 contains tetramer. That means there are 4 subunits. Let me draw in a one molecule. So this molecule PFK1, phosphofructokinase 1, has a substrate binding site. Let me draw is S for substrate binding site. The substrate is fructose 6-phosphate. There is also another substrate. It is called ATP. So let me draw. So ATP also has a pocket to bind with this PFK1. But an interesting fact is the allosteric inhibitor is also ATP. So there will be a inhibitory site, allosteric inhibitory site of ATP. Let me draw here. So this is the inhibitory site. Here also ATP can bind and inhibits the enzyme. So in a sense we can summarize that in the PFK1, means phosphofructokinase 1, there are 2 ATP binding sites, one for substrate and the for inhibitor. Now how ATP are actually regulated to bind with this substrate, not to bind with inhibitory region? Yeah, there is a reason. The Km value of this binding is different from this site and this site. The Km value of the substrate is low. That means it has high or higher ATP affinity, and this site has high Km value. That means it has lower ATP affinity. That means when the concentration of ATP increases, then ATP can bind. Okay, you have to understand this. Now another fact I want to mention here, that there is another inhibitory site. Let me draw here. Let's suppose this is an inhibitory site like citrate. Citrate is a product of TCA cycle, so it can also inhibit this enzyme. Now let me draw the activator sites in a green. So this is the activator sites like ADP can activate, AMP can activate activator sites. Then another activator is like fructose 2,6 bisphosphate. This is also an activator, binds with the allosteric activating site. Now the ADP and AMP can activate. So you can understand in a way that when the cells have high level of ATP, this enzyme will get inhibited by the ATP, because there is no need to make ATP if there is high amount of ATP. Now if the cell have low amount of ATP, that means it has more AMP and ATP, then this enzyme can be activated and it can convert fructose 6-phosphate into fructose 1,6-bisphosphate, and the glycolysis process can go on. And can you see that this enzyme has so many controlling site? That is why it's called principal limiting enzyme. Now I will show you another another information for your preparation, that there is a cell's energy charge. It's called energy charge. How to calculate the energy charge of a cell? The concentration of ATP plus 0.5 concentration of ADP. If you divide this with the total concentration of ATP plus ADP plus AMP, what you will get is the energy charge of a cell. And this ratio actually determines the function of this PFK1. If cells has low amount of energy or ATP, then this enzyme will be activated. If the cells have more energy charge, then it will not activate it. Now another thing I have mentioned, the very important point you have to remember, that is the fructose 2,6 bisphosphate. How this molecule fructose 2,6 bisphosphate, you can see this is the fructose 1,6 bisphosphate. Here first carbon phosphorylated, 6 carbon are phosphorylated. Now another way this second hydrogen bond can be phosphorylated and the 6. So this is called fructose 2,6 bisphosphate. How it is actually produced and how it actually activate this PFK1, I will show you here. This is the branching of this glycolysis process. Fructose 6 phosphate can be converted into fructose 2,6 bisphosphate. Do you know where this thing occur? In the liver, if the glucose concentration increases, then the fructose 6 phosphate can also be converted into fructose 2,6 bisphosphate. The enzyme name is, the name of the enzyme is PFK2, Phosphofructokinase 2, and it also use ATP and make ADP and can phosphorylate in the 2 position of the fructose. Okay. Now the reason here the regulation is that this molecule fructose 2,6 bisphosphate can activate this enzyme. Fructose 2,6 bisphosphate has a binding site, allosteric binding site, activating site in the PFK1 site, and it can decrease the inhibitory power of ATP. So this is another mechanism of these regulations. Now I will tell you another important information, that insulin positively regulate or activate this enzyme PFK2. Insulin positively activate the PFK2, but a opposite enzyme called glucagon can inhibit this enzyme. Okay, so this is the third step of glycolysis, you can remember, and I am showing all the informations for each and every step. In the next class we will learn the fourth step, or step 4 of the glycolysis. So thank you for watching, and always keep learning.